but today I'd like to share some of the work we made recently about the material property of microtubules and submit those observations to your expertise as a material scientist. So microtubules is this network that you can find in cells. So these are long polymers. Their length is about the cell size. They are nucleated by the centrosome, which is located here, that nucleate those filaments that grow up to the cell periphery. Those filaments are highly dynamic, permanently growing and shrinking, and they are the support. They serve as tracks for molecular transport in the cell. So somehow the architecture of this network will determine where the cell will secrete some factors or where it will engulf and collect external factors as well. So it somehow determines the entire cell polarity and its orientation in space. We were interested in the architecture of this network, and to study it in controlled conditions, we plated cells on macro patterns. And when you do so, you can see that it has the remarkable property to self-center. And so the architecture of this network and somehow the balance of mechanical forces self-position the centrosome, the center of the aster, at the exact center of the cell. And this regardless of the shape of the pattern that you can use. And if you look at different cells, you can see that this is a very robust property. You see this network is highly asymmetric. You have more microtubules here than here, but it is still positioned at the center. In this other cell, you have a mixture of different shape of microtubules. Some are long and regular curvature. Some are much more curvy. And despite the diversity of microtubule shapes, the network can still find the cell center. We were intrigued by these observations and made some numerical simulations about what could be the forces in the network allowing it to self-position at the cell center. What has been proposed is that there are some molecular motors everywhere in the cytoplasm that are pulling on those microtubules, pulling them under tension. And of course, if you have regular distribution of tension, the balance is at the center. So if you make some numerical simulations of this, you can see indeed that the network can self-position at the center. But if you look at the shape of the microtubules, they have very regular curvature, which does not correspond to some of the curvature that you can see in cells. So you have a good centering, but the wrong architecture. If now you consider the pushing forces that those microtubules could exert on the cell periphery, of course, these pushing forces can be different depending on the stiffness of your microtubules, if they are soft or stiff. We compare these two behaviors, and here you have stiff microtubules, and here you have soft one. And the centering is less good, because here, as soon as some microtubules will reach this part, they will prevent the motion of the centrosome toward the center. When they are soft, the curvature that we see is much closer to some of the observations that you can make in cells. But you lose the centering ability as well. So it seems that this centering capacity is not so obvious, and we don't understand how it works. And it obviously, what is the most difficult to get is the real architecture of this network. How this architecture is able to self-center. It's probably a question of material property of this network to get the good shapes and the good mechanics. And currently the current model can't explain this behavior. So I think that to further understand the mechanics of this network, we have to go to the fine structure of microtubules. So on this picture, you can see those microtubules in this drawing. And you can see that these are not cords or single filaments. These are tubes. Okay, this is 25 nanometer tubes. It's a hollow tube. And this structure here, so these are made of protofilaments. So here you have 13 protofilaments next to each other. And those 13 protofilaments form the tubes. And basic considerations suggest that the shape of these tubes is quite important in the stiffness then of the microtubules. And because these are hollow tubes, it has been shown a long time ago by Galileo that if you have a given amount of material, whether you make a full tube or a hollow tube with the same amount of matter, you're going to make some more rigid tubes when you have hollow tubes. So this architecture is important, and it confers some high stiffness to the microtubules. Actually, microtubules are the stiffest organelle in the cell. So it's important to measure this mechanical property. But it's very difficult in cells. How can you measure the stiffness of microtubules in cells? Something you can do is to work out of the cell. Purify all the components, put them on a glass slide, and look at their physical property. So what we did is that we purified centrosomes. So you open the cell, release everything, and then collect all the centrosomes that are the nucleating center here, and you put them on your glass slide. On the side, you purify the tubulin monomer that forms the microtubules. And when you add the tubulin to your centrosome, those microtubules, they can grow on your glass slide. And you have all the dynamic properties of growing and shrinking. But in this case, the centrosome has only been, they fall on the slide. And if you want to, but they can move everywhere. So it's difficult to apply some forces because you need to apply some forces on it to measure the stiffness. So what we did is that we took advantage of our micropatterning methods in order to graft some very short microtubule piece on this array of dots, so that now this part of the microtubule here is attached to the substrate. The substrate here is passivated with polyethylene glycol, so the microtubules can grow, and as you can see, they can move. They are attached only by their minus end, and the rest of the microtubule can bend and move. And you can vary the shape of the nucleation that you want. Here some bars are quite convenient. You will see later why. And we can combine this with a macrofluidic device in order to apply some hydrodynamic forces on the growing microtubules. This is how it goes. You have your micropattern substrate, on top of which you have your macrofluidic device. You bring some seeds, so the very short microtubule seeds, so that they can attach to the bars. Then you add the free tubulins so that your microtubules can elongate, and because of the orientation of the flow, most of them are perpendicular now to the bars. And then you take advantage of the other channels in order to create some flow and bend your microtubules. And as you bend the microtubule, so this is how it goes, you have red beads to measure the flow. If you know the flow, you know the hydrodynamic force. If you measure now the shape, if you know the force, you know the shape, you can estimate the rigidity of your material. And we repeated the experiment to have a sort of better measurements. And what we saw, and that you may probably have seen here, is that by these repeated cycles of bending and release, these are the projections of the microtubule behavior as we repeated the bending cycles. And what happened is that they deform more and more. They were quite stiff at the beginning and much softer at the end. So those microtubules, what we found is that they soften under mechanical constraint. And this can be seen on this graph. So in the field, people use the persistent length. It's a measure of it's the bending stiffness, actually. And we normalize it with respect to the first bending stiffness. So the persistent length here is about 5 to 10 millimeter, meaning that it takes 10 millimeter for the two ends to be completely decorrelated, which is an order of magnitude bigger than the cell size. Two order, actually. And so anyway, what is important is that you can see here that all the red curves show the microtubules that have significantly softened after the bending cycles. Few of them, the green one, did not and maintained their stiffness. So this is not something that is intrinsic to all microtubules. Some do soften, some don't, which is surprising. But material scientists told us that is completely normal. Every material submitted to repeated cycles of bending will undergo the fatigue process. And And after time, it will damage after every cycles of forces. And this is due to the fact that the material is not perfect. There are some defects in the material. A good crystal would deform elastically like this and recover perfectly its shape. But if there are some defects in your crystal, then as you bend it, you will accumulate some stress on the defect and you will damage your structure. And actually, some defect has been reported on microtubules. Here you have some AFM pictures, so you can see the protofilaments, but you can see also some holes that can happen from time to time in the lattice. Here, of course, and even sometimes some huge cracks. But you can think that the AFM made the holes. But not only with AFM you can see some damage. What is quite frequent, if you make some EM pictures of your microtubules, here the protofilaments are very clear, and then when you make cross sections, you can average that and measure the exact number of protofilaments in a given cross section. In this part of the cross section, here you can measure 13 protofilaments. But in this one here, you have 14. So it can vary, but it means that at the transition between 13 and 14, you have a structural defect. And these defects are quite frequent. Every 1 to 10 micron on the microtubules, you have those defects. And what has been shown by this team is that the speed of assembly of your microtubules controls the amount of defect. If it grows slowly, at low concentration of tubulin, you have very low rate of defect. But if they have been assembled fast, then you have a larger amount of defect. And the speed of assembly can be controlled by the concentration of the free monomer. So we varied those concentrations to make microtubules without or with defect. Then we bend those microtubules. The ones that have no defect were stiffer than the one that had some defect. And more interestingly, when the structure was perfect, you see, those pictures show you the maximal deformation cycle after cycle. It overlaid completely, it's white, meaning that it was perfectly elastic. And when you had some defect, then you had the softening process I described before. So the defects were responsible for the softening process of the microtubules. Interestingly, we performed this other experiment. We repeated the cycle of bending. You have here 5 cycles, so the microtubules can get softer and softer. The lattice has been damaged. And then we waited. We waited 100 seconds. And what happened during those 100 seconds? The microtubules recovered their stiffness. So this is classical for any type of inert material. When you apply forces, they damage, they get softer. But this is a unique property that has been described only for this type of living matter. It can self-heal, repair, recover initial mechanical properties. How can this happen? We suggest is that when you have some defect, apply some forces, accumulate stress on the defect, lose some dimer, it becomes softer. Then you have a bigger damage. It was a small defect, it has become bigger damage. But this is a self-assembled material. If you still have some dimers around, they can come back and heal the structure. To test this hypothesis, we took advantage of our device in order to distinguish the color of the microtubules when they have been assembled with some red fluorescent tubulin. And then we bend those red microtubules in the presence of free green tubulin, so that if any of the surrounding dimers come to heal the material, we're going to have some green tubulin healing the red microtubule. So this is how the experiment goes. You have your red microtubules and you bend it in the presence of surrounding green dimers. And we repeated the cycles of bending. Then we waited. After that, time will be accelerated. And we wash out. And what we saw, and that can even better be seen on those fixed images, is that the microtubules keep on elongating because these are dynamic polymer, but they also incorporated some external tubulin in their lattice. So these were the proof that you have some incorporation along the length of the lattice, which has never been described before. What was known was this part. The end are dynamics. This has never been described before. This is the self-healing of the material. That was the conclusion made by Laura and Karin. So Laura was a PhD student, now looking for a postdoc. And Karin, she's a physicist, theoretical physicist working with us. They conclude that the microtubule bending can damage the structure of the microtubule, making it softer. But the damaged microtubules can self-repair by incorporating new dimers in the lattice. So as I said, this completely changed our view of the microtubules. What was believed before is that the only dynamics occur at the tip of the microtubules where it grows and shrinks. What we propose is that there are also some turnover along the lattice. What has been done in our case was to submit those microtubules to physical forces. And we further wondered whether these dynamics could occur without any external force. How intrinsic is this new property to microtubules? Is it only under mechanical forces, or does it happen all the time in all microtubules? So the problem is that the difficulty actually is that microtubules in vitro are highly dynamic, more than there are actually in cells. And we had to study this dynamic, which is probably a slow process compared to this one. We had to block this exchange here in order to interrupt the possibility of depolymerization, to look only at this process. So to do this, we grew microtubule here, and then we add what is called a cap. It's a form of tubulin that can't be hydrolyzed because tubulins are bound to some GTP. And this nucleotide, when it hydrolyzes, it allows the depolymerization. But if you have some non-hydrolyzable form of tubulin, you make a sort of part of the microtubule that can't be disassembled. So the microtubule will survive for long. Then we switch the color of the tubulin, and we waited to see whether we could see or not some incorporation in this microtubule. And indeed, we saw some incorporation. So this is your red microtubule, and you see here, we saw some incorporation, although the microtubule was not growing, nor shrinking, no mechanical forces, nothing. Within 15 minutes, we could see the incorporation here of surrounding dimers. And these are various examples. Here you see always the red microtubule and the green. And in most of the microtubule we looked at, we saw some patches of incorporation, you see, along the lattice. And what is interesting is that those patches were quite big, 1 to 3 microns here. And it was not regularly distributed all along the microtubules. There were some patches, okay? And so we tried, as biochemists, we tried to vary the concentration of the surrounding microtubules to see whether higher concentration of soluble tubulin could change the proportion of these patches. And indeed, when we increased the concentration of the green tubulin, we saw that the distance between the patches was reduced, and so we have more incorporation per microtubule. So it's a real biochemical exchange between the microtubule and the soluble pool. If there is actually some turnover, you have some dimers coming in, but you need to have some dimers coming out. But it's much more difficult to visualize the loss of dimers. And the way we did it is to completely remove the dimers around. So if you wash out the free tubulin, let's see if we can start now, if the microtubule will start losing dimers. And so we grow the microtubule. Here is our microtubule. We waited. 13 minutes later, nothing happened. The same microtubule. But then, in the following minutes, it starts weakening in some part here, and then bend, cut, disassemble. Not from the extremity, as people have described before, but from the central part of the microtubule, showing that it actually lost some dimers in the lattice. So those dimers can spontaneously pop off and come back. So we made some numerical simulations to see how likely it is to have those exchange without any external source of energy except the thermal energy. So the energy of the interaction between the dimer in the lattice has been estimated based on the dynamics of the microtubules to be around 30 kT, more or less. That's the total force, or total energy of interaction with neighbors. And so we made those simulations with this type of value. And here it's how it will evolve over time. So here's the microtubule at the beginning. And what has been colored here is the number of incorporated dimers per section. So in a given section, you have 13 protofilaments, okay? And we waited, and this is over time. You see that you have progressively, few points, and then many. And then you see that if we put those value of 30 kT of interaction for every dimer with the neighbor, we should see three dimers per section. It's a third of it or a fourth of it, which should be labeled. So we should see completely red microtubules all along the length after 20 minutes. That's not what we saw. But what is interesting is that if we switch now to 40 kT, because it was an estimation, nobody has been ever able to measure those energy of interaction of dimers. Now it's much less frequent. What can be measured here is that we have, it's not all the cross sections. You see here it's 10 times less, and it's not all along. Well, it's the good frequency now that we can observe, but it is distributed all along, which is not what we saw. We saw patches. How can we get patches for something that could occur everywhere? Probably the key for that is what I mentioned before, is that you can have some defect in the lattice. Those defects, when you have a defect like this, of course the energy of the interaction for those dimers is much lower because there are some missing one. So of course for this one, or this one, it's easier to pop off because it has less interactions. So we took into account the fact that there were some holes. In this example, we put some holes here and there. And of course, these are the preferential location where the incorporation occurs. And now we could see some patches with the right frequency and the right size of patches, suggesting that those holes, those defects, were the source for the turnover of the dimers in the microtubules. So we tested this hypothesis by varying the amount of defect, because as I explained you, we can control the amount of defect. We can grow microtubules here with very low defect number and some with higher defect number. And you compare these three pictures. In this one, you can see that has lots of defect. We saw lots of incorporations, whereas in those ones, it was much less frequent. And this has been measured on this. You see here, 0.2 incorporation per micron, and here 0.06. So I don't know how precise are those values, but it is clearly depending on the amount of defect that you have in your microtubules. So the defects were probably the source for the turnover of these dimers. And this is the main conclusion of our work, is that what has been proposed is that microtubules are, the tip of microtubules are dynamics. But we think that the entire lattice is dynamic, permanently exchanging dimers, as it is shown here. And that's an interesting property in terms of materials. It is due to the low energy of those interactions. These are not covalent bonds. These are electrostatic and van der Waals and hydrogen bond interaction between the dimers. So it's a soft materials. And because of this low energy of interaction, the material can exchange with the soluble pool. So it's a material that is permanently renewing itself. And this provides some interesting property to it. It can self-repair, and by self-repairing, it can adapt to external forces. And if it can adapt, it can optimize. And if it would be a perfect lattice, it would not occur. So that's also an important message that we got from this work, is that if you have a perfect lattice, it's boring. Nothing happens. You have these dynamics, but you have some very fixed material property that you have to cope with. If you have some structural defect, life is much more fun, because then the first thing you can do is that you can enlarge the range of stiffness. Depending on the rate of defect, you can make some soft or some stiff microtubules. And also, what I did not discuss here, and that will be the focus for future work for us, is that it can also enlarge the diversity of biochemical modification that can occur all along. Because there are some proteins that have some specific affinity for the new dimers. Here what I discuss is that those are the GTP bound dimers, and these are the GDP bound. They have lost some energy, okay? But some proteins are specific for these guys. But now these guys are not only at the tip, they are located all along. So if you have any proteins that have preferential binding for the microtubule tip, for the microtubule GTP, it can be present all along the lattice. If you have some specific enzymes or microtubule-associated proteins, they could also take advantage of these holes to interact with the lattice, and why not within the lattice? And of course, it has been shown that some enzymes, like severing enzymes also, can act on microtubules. And for those enzymes as well, it could be very interesting to have some structural defects, some local weakness they took advantage of to cut microtubules on specific locations. So, and by repairing, of course, you add new dimers, and by adding new dimers, you could generate new microtubules. Why not new branches? So, what I want to say is that by self-repairing, you can adapt under stress. If you have some defect, then you can generate local high curvature. You can self-repair. As you repair, you can make new branches. And when you have defect and you apply some pushing forces, now the way you bend will be like this and not with the homogeneous curvature. That changes a lot the propagation of forces through the network. And the question we want to ask now, and that we will focus on in the next future, is that if we now take into account all this diversity of mechanical behavior of microtubules, will we now be able to explain these weird shapes of microtubules that we saw, and how the associated properties are compatible with this self-centering of the microtubule network. With this, I will thank the whole team that is working on actin or microtubules. And we have an open postdoc position to work on microtubules in vitro in Paris with us. I'd like to thank our financial support. And this was part of our work dedicated to microtubule, but we are also working not only on microtubules, but also actin filaments and cell shape, cell polarization. And if you are interested, you should have a look at our website to know more about this. Thank you for your attention.